Hello, my name is Martina Kutmon and I'm an assistant professor at Maastricht University. Today I will talk a little bit about the COVID-19 work within the WikiPathways project. As one of the WikiPathways architects, I would like to start with an acknowledgement page because there are a lot of people involved and the work that I will present today. Together with Alex Pico from the Gladstone Institutes, I'm leading a team of core developers and admin curators, but most of the content and many of the features come from contributors and pathway curators around the world. Most of you probably know about WikiPathways, but I will give a short introduction of the fundamental concepts of our resource before highlighting our COVID-19 specific work. WikiPathways was launched in 2008 as an experiment in community-based curation of biological pathways. Already back then, there were simply too many publications that it was difficult to keep the knowledge up to date, accessible, and integrated. And that's even more true nowadays. So building on the idea of crowdsourcing, we want to take advantage of the direct participation of expert researchers. WikiPathways is built on the same platform as Wikipedia, so we focus on collecting and curating knowledge about biological pathways. The database is community curated and everybody can freely contribute, edit, and use the pathway collection. This provides a platform where new findings can be added immediately, ideally by the experts themselves. Discussions about content is really encouraged, and version history allows easy reversion to a previous version if it's needed. We have successful collaborations with other manually created pathway databases like Reactome or NetPath to provide direct feedback from the community. Using the concept of quality tags, and these are kind of annotations which give information about the creation status of a pathway, we allow users to share and collaborate on pathways still under development, which will not be included in downloadable collections for analysis. Using ontology tags from the disease and cell type ontology, we also allow users to upload disease and cell type specific pathways. On WikiPathways itself, every pathway has its own pathway page with the title, author list, and clickable diagram. Each element is annotated with an identifier, and if you click on the element, cross-references are provided by BridgeDB. Additionally, you can also find the mentioned quality and ontology tags on the pathway page, providing insights in the curation status and the biological context of the pathway model. In the pathway itself, each element, so the nodes and the interactions, can be annotated with publication references, and a bibliography is also provided on the website. As mentioned before, a wiki system keeps record of all revisions of the pathway in the history, and previous versions and differences can be checked and assessed there. Every pathway also has a discussion page, and the pathway can be downloaded in many different formats, from machine-readable formats for analysis to different types of image formats. There are also several ways to access WikiPathways data programmatically. We have a REST API, and our data is also available in RDF format. Additionally, there are different packages and apps for access from R, for example, or Cytoscape. And our human pathway collection is also accessible from the Network Data Exchange, so Index, and Wikidata. And recently, our pathway collections have also been added as gene sets into MSigDB for gene set enrichment analysis. A little bit of a highlight, our SPARQL Explorer recently received a major interface update over the last month using Snorkel. So users can now start from a large set of example queries for data integration, federated queries, and data curation. Additionally to the larger species-specific pathway collections, we also enable communities to create and maintain their own focused collections. So these are the so-called community portals. We currently have over 15 different portals for different communities, from plant portal to rare diseases to COVID-19. The pathway curation work related to COVID-19 is organized in our COVID-19 community portal. This portal currently hosts 34 pathways, which are curated or contributed by over 30 different researchers. But we're actually not working alone on this. The pandemic brought together over 200 researchers from around the world to collaborate, discuss, and work together to build the COVID-19 disease map. In this map, we aim to provide an overview of the complete molecular mechanisms, from the virus attachment and entry, to the virus replication cycle, to the downstream effects in the host, including innate and adaptive immune response. If you click on one of the pathways, you can zoom in and see pathway models, and they're provided in many different formats, including SPGN and SBML, so that they can be used for data visualization, analysis, and modeling. Obviously, this map is still under development, and in the weekly telcons, the curation efforts are aligned and streamlined between the groups. The important part of this community is that it brings together researchers from many different disciplines, including the databases and resources, the tool developers, the data producers, text mining, computational modeling, and the data analysis part. One of the main aims of the COVID-19 project that I lead is the development of continuous reproducible data analysis workflows. This way, it's easy to rerun an analysis if there's a new pathway coming up or a new experimental data set is available. We use and further extend on our existing pathway and network analysis workflows. The workflow that I will present today is implemented in R as a Jupyter notebook, and it uses transcriptomics data to study the molecular mechanisms affected during the COVID-19 infection, as well as highlight still missing information in our disease map. I will present the results for a publicly available dataset by Blanco Melo et al., where they exposed two different epithelial cell lines to a mock and a SARS-CoV-2 virus for 24 hours. After the differential gene expression analysis, we can see a stronger effect in the NHBE cells, so the human bronchial epithelial cells, and so I will only discuss the results for this cell line today. We then performed an enrichment analysis using an overrepresentation analysis and identified 45 altered pathways. The majority of these pathways involve some immune and signaling processes that are potentially related to the host response to the virus infection. There are several pathways out of the COVID-19 specific collection, including the type 1 interferon pathway shown on the right, and the coagulation pathway. Once we have the data, we can now visualize it on the pathway model. We can see how the different genes are reacting to the virus infection and can study the molecular mechanisms in detail. But importantly, these pathways are not independent. They often overlap with each other. So if we visualize this relationship between the pathways in a pathway chain network, it allows us to actually study the crosstalk and interactions between them. We can then also extend this network with drug target information from DrugBank in Cytoscape, and 444 known drugs were added to the network. Interestingly, 245 of these are FDA approved, and several drugs that we found were also listed as experimental unapproved treatments for COVID-19. For example, tolisizumab is an interleukin-6 receptor inhibitor, which is known to treat cytokine release syndrome and has, in several clinical trials, also shown possibly positive effects for COVID-19 patients. From a curation perspective, it's also important to identify what we are still missing in our large disease map. So we used the protein-protein interaction network approach, where we used all the differentially expressed genes and created a network using the string database. And then we added the information about the pathways in that network. So we now can see which genes are present in COVID-19 specific pathways. There are, however, a lot of affected genes which are not present in any of the pathways, so they're not connecting to any of these green nodes in the network. So analysis like these help us to target the curation efforts. We're currently also running this workflow on many other COVID-19 transcriptomics data sets that allows us to then further prioritize curation and analysis efforts. Now to shortly summarize. WikiPathways is a community-curated pathway database, and the COVID-19 pathways on WikiPathways are curated by many different researchers. The successful collaboration with the COVID-19 disease map project really accelerates correction efforts and allows more in-depth analysis using highly curated pathway models. The automated analysis workflow presented today is able to identify affected pathways that are changing after COVID-19 infection. It allows researchers to study how pathways are connected and which drugs are already known to target interesting genes and target candidates. Lastly, it also allows us to highlight which genes are affected in an infection, but for which we do not have any further detailed molecular mechanisms in the disease map yet. We're also looking into integrating other types of omics data, including proteomics and metabolomics, to get a more complete view of the response of the virus infection in these pathway models. I would like to thank you for your attention, and I'm happy to answer any questions you might have.